Hello friends, a very good morning to all of you. YouTube चैनल पे आपका हार्दिक अभिनंदन। आज मैं आप लोगों के पैथोलॉजिकल यूरिन जो कि एबनॉर्मल कंस्टिट्यूएंट ऑफ यूरिन के अंतर्गत आता है के बारे में बताने वाला हूं। जैसा कि आपको मालूम कि यूरिन में पैथोलॉजिकल कंस्टिट्यूएंट्स ऑफ यूरिन के अंतर्गत ग्लूकोज, कीटोन बॉडीज, प्रोटीन, बाइल सॉल्ट्स, बाइल पिगमेंट्स एंड ब्लड है। तो आज मैं आप लोगों को कीटोन बॉडी के बारे में विस्तृत रूप से बताने वाला हूं। फर्स्ट ऑफ ऑल, we know what is ketonuria. Ketonuria means presence of ketone bodies in urine. Property of ketone bodies. Ketone bodies are water soluble molecules that contain the ketonic group produced from fatty acid by the liver. And the process name is ketogenesis. Formation of ketone bodies is known as ketogenesis. They are radially transported into tissue outside the liver, where they are converted into acyl coenzyme A, which then enters the citric acid cycle. The peculiar thing about ketone bodies is it can cross blood-brain barrier directly into brain during shortage of glucose. Ketone bodies are metabolized in brain easily. Ketone bodies and its origin. There are three types of ketone bodies: acetone, acetoacetate, and beta-hydroxybutyrate. Liver is the exclusive site for ketone bodies. During low glucose concentration, during prolonged starvation, and uncontrolled diabetes mellitus, the formation of ketone bodies takes place. Ketone body synthesis, breakdown, utilization, and its biological importance. Liver only synthesized but cannot utilize ketone bodies. It is utilized by peripheral tissue during depleted glucose level. कीटोन बॉडीज का सिंथेसिस तब होता है जब शरीर में ग्लूकोज की मात्रा कम हो जाती है। कीटोजेनेसिस ड्यू टू इंसुलिन डिफिशियंसी in case of type 1 diabetes mellitus, less amount of glucose uptake by the cell. Hence, glycogen stores typically are depleted within the first 24 hours of fasting. What is ketogenesis? Synthesis of ketone bodies is known as ketogenesis, and the exclusive site for ketone body synthesis is liver. Ketolysis is breakdown of ketone bodies, and this ketone body is utilized in muscle and brain. Ketonuria means presence of ketone body in blood. Ketonuria, presence of ketone bodies in urine. When the renal threshold limit for ketone bodies cross 3 mg per dl, then it appears into the urine, and this condition is known as ketonuria. Biological importance. Ketogenesis is used to get energy by the brain, heart, and skeletal muscle under fasting condition. Clinical condition, pathological condition in which ketone bodies come into the urine. आखिर में कौन कौन सी क्लिनिकल कंडीशन है जिसमें कीटोन बॉडीज का प्रेजेंस यूरिन में देखा जाता है? Diabetes ketoacidosis and ketoacidosis. These are clinical conditions, whereas prolonged starvation, which comes under physiological condition. Vigorous exercise also leads to production of ketone body in urine. So these all are clinical as well as physiological conditions in which ketone bodies appears in urine. What are the biochemical alterations in diabetic ketoacidosis? Diabetic ketoacidosis is associated with severe high blood glucose, insulin deficiency, and a disruption of the body's acid-base balance. Ketonuria and glycosuria causes increased urination, thirst, dehydration, and a loss of electrolytes. Symptom: rapid breathing, shortness of breath, a fruity scent to the breath, nausea, vomiting, fatigue, confusion, and eventually coma. Next, what is ketoacidosis? Explore Kussmaul's respirations. Name the clinical condition in which Kussmaul's respiration observed. Ketoacidosis are normally buffered in tissue and blood. Shortage of glucose inside the cell leads to production of ketone bodies. Excess amount of ketone bodies affect alkali reserve by depleting buffer cation, which in turn cause ketoacidosis. Diabetic ketoacidosis patients have a typical breathing pattern known as Kussmaul's respiration, which is a compensatory hyperventilation. Fruity-like smells appear in diabetic ketoacidosis patients due to the presence of acetone, and acetone is the ketone bodies which is volatile in nature. Blood ketones are primarily used to screen for, detect, and monitor a serious, sometimes life-threatening condition called diabetic ketoacidosis in people with type 1 and sometimes 2 diabetes. Lab test for ketone bodies in urine. Here we discussed about Rothra test. The requirement is powdered ammonium sulfate, 5% sodium nitroprusside, and third one is concentrated aqueous ammonia. Procedure of Rothra test. Take 10 milliliter of urine sample of patient and a pinch of solid ammonium sulfate and shake till the solution becomes saturated. Add 2 to 3 drops of freshly prepared sodium hydroxide and mix gently. Add 1 ml of strong ammonium hydroxide along the side wall of the test tube. What is the absorption? A reddish brown ring develops at the interface of the two liquids, indicating the presence of ketone bodies in the test tube. The reaction of the test is nitroprusside in alkaline medium react with a ketone group to form a purple ring. It is given by acetone and acetoacetate but not by beta hydroxybutyric acid. Nowadays, reagent strip for ketone bodies are commercially available and are simple to use. Thanks for watching my video on YouTube. Subscribe my channel for more update. Thank you. Have a nice day.